I'm going to talk you through the answer to question C5.1. Okay, so we'll have a look at the first bit. Flowering in many plants will only take place when the photoperiod, that is the relative length of day and night, is appropriate. It is controlled by a photoreceptor pigment. Name the photoreceptor pigment which is involved in the control of flowering in plants. Okay, so for A part 1, we're looking for phytochrome as your answer. Okay. If we look at part 2, in which part of the plant is the photoreceptor pigment located? Okay, and the photoreceptors are located in the leaves of the plant. So we're going to move on, and in the next bit of the question they've given us a little diagram. Okay, so the outcome for flowering in chrysanthemum plants when exposed to 4 different light regimes is shown in the diagram below. So if we look at the little graph, it's split into sort of, we've got the time along the x-axis. So the black is representing the darkness and the pale section of the graph is representing the light. Okay, so if we look at regime 1, in regime 1 they have given the plant a short period of darkness and a long period of continuous light, and we have no flowering for the plant. Okay. In regime 2 we have a slightly longer period of darkness and there's still no flowering. Okay, and if we look at what that period of darkness there is, it's about 12 hours. Okay. Look at regime 3, you've given it a longer period of darkness. This time we've gone more than 12 hours, so it's exceeded the critical length, and we can see then that flowering is stimulated in the plant. Okay. If we look then at regime 4, we have a similar period of darkness, but it has been interrupted by a flash of light, so it's not continuous darkness, and there's been a flash of light in between, and as a result of that we have no flowering occurring in this plant. So the question is asking us, using the information in the diagram, determine whether the chrysanthemum is a short day or a long day plant. Okay. So if we have a look, short day plants will only flower if they have a long period of uninterrupted darkness. Okay, and that's the case for this plant. So this plant is a short day plant. Okay, now we need to go on to kind of explain why. Okay, so you're going to mention, so you get 1 mark for mentioning that it is a short day plant, and another mark for saying it requires a long period of uninterrupted darkness, and then we need to mention the time frame here, that is greater than 12 hours of uninterrupted darkness. Right, in part 4 then, using your understanding of the phytochrome system, explain the difference in the flowering outcomes in light regimes 3 and 4. So we want to know why there is a difference between these two. Okay, so they've both got the same length of daylight, if you like, but this one has been interrupted by a flash of light. So if we think about the photoreceptor pigments first, so I'm going to, if we diagram here, okay, so we have P660 and we've got P730. Okay, P660 will get turned into P730 during sunlight or by being exposed to red light. Okay, the P730 can be turned back to P660 in the darkness, but it's very slow. If you expose the plant to far red light, then it will be a quick conversion. Now, what we need to know about P730 is that P730 inhibits flowering in short day plants. So if they're going to flower, they need this to be removed. So in nature, during like the winter, sort of spring, autumny kind of time, when you have long nights, we've got a long period of uninterrupted darkness, this P730 can gradually be removed. Okay, if you remove enough of it, the plant will flower for you. Okay, so if we're looking at what's happening, okay, we've got to explain the difference between regime 3 and regime 4. So in regime 3 we have a long period of uninterrupted darkness. Okay, that allows the P730 to be removed. Okay, and P730 inhibits flowering in short day plants. Okay, and the conversion in the darkness is slow. Okay, if we look at regime 4, so during the darkness, very gradually you're removing this and allowing P660 to build up. Okay, if you break that darkness with a wee flash of light, whatever you have made in terms of your P660 will rapidly go back to P730 because the conversion of P660 to 730 is fast. So a wee flash of light will take your 660 and turn it back into 7:30. So all the work that's been done by the plant during the night, removing this, is undone. Okay, so if we're looking at regime 4, this flash of light will rapidly convert the P730 back into the P660 back into P730. Okay, so whatever 660 they've built up will be converted to 730, which we know inhibits the flowering. Okay, so I'll just show you the mark scheme answer for that. You can pause it if you like. Okay, so the marks were broken down. Flowering in short day plants is inhibited by P730. In regime 3, the dark period is long enough to allow P730 to be converted to P660. Okay, lack of P730 will allow flowering to occur. And then regime 4, the flash of light converts any P660 back to P730, so there won't be any flowering occurring. The next wee bit of the question relates to hormones and plants. So if we have a look at the little diagrams, we've got 2 plant hormones, A and B, applied to decapitated shoots. The effects of each hormone after 5 days on the relative size of the cells within the shoots is shown in the diagram below. Okay, so we want to know for part 1, describe the effect of each hormone and identify the hormones. So if we look at this one, this has got hormone A on it. So it's a little block with the hormone. Here's the before, here's the after. We haven't got any more cells, but the cells that we do have are elongated. So what we're looking for here is that hormone A is auxin, and our reason is it promotes cell elongation. Okay. If we look then at hormone B, this is our starting point, this is our finishing point, and we can see we've got loads more cells. So this hormone has encouraged cell division. So this hormone is cytokinin, and your reason is that it stimulates cell division. Okay. Now, part 2 on this B part 2 suggests an appropriate control for this experiment and explain why it is necessary. Okay. So as well as applying the hormone, it's in agar. So agar is like a little jelly. So the hormone is in agar. You need a control that would be a block of agar with no hormones in it, and the reason for that is, well, perhaps these changes have been brought about by the agar and not the hormones. So if you use a block that is just agar with no hormones, it allows you to make a comparison between the two. Okay, thank you.